Hey guys, welcome back to my channel. This is Debantana and you're watching Eduplex. So this video will basically comprise of a part of sexual reproduction in flowering plants. First of all, as we are talking about sexual reproduction, we have already discussed about what is sexual reproduction, phases, types of reproduction and many such primary facts to be known. Now here we'll be talking about the details about the types of sexual reproduction. Let us recapitulate some of the important facts from our last chapter. So what do we mean by sexual reproduction? Basically, sexual reproduction involves the formation and fusion of gametes formed either by the same individual or by two different individuals. So in this video, we'll be talking about the flowering plants. So basically we'll be discussing about the structure of flowers, the male and female reproductive organs, what are flowers and the steps required to conduct sexual reproduction in flowering plants. So starting with flowers. The flowers are different from the outside, but internally the reproductive organ are mostly same in all flowers. Basically, a flower is a shoot modified for reproduction. It is the fascinating organs of angiosperms. Many hormonal and structural changes are initiated, leading to the differentiation and development of floral primordia. The floral primordia first develop into floral buds and then into flowers. Flowers bear the reproductive organs where gametes are produced. Androecium is the male reproductive whorl. It consists of stamen, whereas gynoecium is the female reproductive whorl. It consists of carpel. So basically talking about the female reproductive organ. The female reproductive organ or the carpel is divided into three parts, that is stigma, style and ovary. Whereas the male reproductive organ, that is the stamen, is basically divided into anther and the filament. Now certain processes are involved for the sexual reproduction to occur in flowering plants. They can be classified into three steps, namely pre-fertilization, double fertilization and post-fertilization. Now we'll be basically talking about the pre-fertilization in this chapter. So basically this is a huge topic to be discussed. In this video we'll be primarily concentrating on the pre-fertilization step of sexual reproduction. This brings us to the fact that what is pre-fertilization? We know what do we mean by pre-fertilization, which we have already learned in the first chapter. So in short, it is that stage of sexual reproduction where fertilization does not occur. That is the step just before the fertilization is occurring. So let's start with pre-fertilization. Basically, pre-fertilization comprises of four stages, that is pollen grain formation, embryo sac formation, pollination and pollen pistil formation. Talking about pollen grain formation, it basically comprises of stamen, microgenesis and pollen grains. So let us talk about stamen. So as I have already said, a stamen is the male reproductive unit of angiosperms. It basically consists of two parts, a long slender stalk called filament and the terminal bilobed structure called anther. The proximal end of the filament is attached to the thalamus or the petal, which is why it is known as epipetalous. So this brings us to the point of structure of anther. A typical anther is bilobed with two theca in each lobe. That is why it is known as dithesis, a longitudinal groove which separates the theca. In a cross section, the anther is a tetragonal, that is four-sided structure consisting of four microsporangia, two in each of the lobes. Later, the microsporangia develop into the pollen sacs, which consist of large number of pollen grains. They extend all through the length of the anther. A microsporangium is more often circular in outline and is surrounded by four wall layers. These layers of anther are formed by Archesporial cells. The Archesporial cells are basically a primitive group of cells that form cells from which spores are developed. That is, Archesporial cells are basically classified into primary parietal cells and primary sporogenous cells. Now, talking about primary parietal cells, these cells divide several times to form the anther wall, whereas the primary sporogenous cells divides to form microspore mother cell, which is denoted with PMCs, that is pollen mother cell. As I've already told that there are 4 layers of anther. Now let us talk about the 4 layers of anther. The first layer is the epidermis. It is the outermost single layer which is protective in nature. Then we have the endothecium. This comprises of cells which develops thickenings and helps in dehiscence. Now we have middle layer, which is 2 or 4 layers of cells. Lastly, we will be talking about tapetum, which is the innermost layer of large binucleate or multinucleate cells with dense cytoplasm which nourishes developing microspore inside. Now this picture shows us the 4 layers or 4 wall layers of the anther. When the anther is young, the microsporangium contains compactly arranged homogeneous cells forming the sporogenous tissue. Now let us talk about the second step of pollen grain formation, that is microsporogenesis and formation of pollen grains. The formation of microspores inside the microsporangia or pollen sacs of angiosperm is known as microsporogenesis. Every cell of the sporogenous tissue is a potential pollen mother cell or PMC, but some of them forego this potential and others become differentiated into the pollen undercells of microspore mother cells. Each microspore mother cell undergoes meiosis to form a cluster of 4 haploid cells called microspore tetrad. As the anther matures, the microspores dissociate from one another from the tetrad and develop into pollen grains. The nucleus of the microspore undergoes mitosis to form a large vegetative cell and a small spindle-shaped generative cell that floats into the cytoplasm of the vegetative cell. Usually, the pollen grains are liberated at this 2-celled stage. In certain species, the generative cell divides mitotically to form 2 male gametes, and the pollen grains are 3-celled during liberation. Now this brings us to the last stage of pollen grain formation, that is pollen grains. Basically, pollen grains are microscopic structures that carry the male reproductive cell of plants. Pollen grains are normally spherical in outline. They develop a 2-layered wall. The 2-layered wall comprises of outer exine and inner intine. The outer exine is hard, thick, smooth and made up of sporopollenin. It shows a variety of architecture characteristics of each species, primarily providing resistance to biological decomposition, whereas intine is thin, continuous and is made up of cellulose and pectin. Basically, the sporopollenin actually preserves the pollen grains for a long period and provides protein for enzymatic and compatibility reaction. No enzyme can degrade sporopollenin. At certain places, exine is either absent or very thin, and such places are called germ pores. A mature pollen grain has 2 cells, a vegetative cell and a generative cell, as I have already discussed. The vegetative cell is larger, has abundant reserve food and a large irregular shaped nucleus which is responsible for formation of microspore involved in syngamy or fertilization. The generative cell is small, spindle shaped and floats in the dense cytoplasm of the vegetative cell. The cytoplasm of pollen grains basically consists of saturated oils and starch. The pollen grains of many species such as Parthenium cause severe allergy and bronchial afflictions like asthma and bronchitis. Now, talking about pollen grains, we should also know the pollen viability, which means the period till when the pollen grains remain viable, that is only for 30 minutes. This also depends on the temperature and the humidity where the pollen grain sustains. Now we have certain uses of pollen grains as well. The pollen grains are rich in nutrients and are used as food supplements in the form of tablets. Consumption of them has been expected to increase the performance of athletes and race horses. So with this, we complete our today's topic, that is pre-fertilization. In the next video, we'll be talking about the embryo sac formation, pollination and pollen pistil interaction. Till then, keep revising.